Yes, now I'm able to hear. Very good evening, Dr. Sargunadevi. Sorry to keep you on hold. I have some technical glitches here. Can we start? Are we live?
Really fine. Yeah, no problem.
Can we start, Sindhu?
Yes, ma'am.
A very good evening to one and all. A warm welcome to all the delegates who have joined today for yet another masterclass in embryology series. So we have been doing regularly monthly masterclass series as part of the SCALE initiative. And we are doing masterclasses both in embryology and advanced ART. So all of us, have had various discussions on various topics in the last couple of months. And just to have glimpses of what we had, we did have master classes from Dr. Sujata Ramakrishnan, Dr. Gaurav Majumdar, Dr. Sandeep Karunakaran, and various other stalwarts in this field, giving us a lot of insights and knowledge into different topics, totally dealing with A to Z of a complete topic. And we did have the entire recording shared with all the delegates who joined in the meeting. And also at the end of the meeting, we also did share an e-certificate and necessary articles related to the masterclass topic. So without much ado, I would like to announce the next masterclass. That will be a follow-up of the masterclass which was done in the month of April, which will be done by Dr. Sachin Kulkarni, that is Advanced ART. We're going to have novel stimulation protocols. Followed by 19th of June, Dr. Kesri Avri will be talking on freezing to thawing, the full cryopreservation journey. So today we have yet another stalwart in the field of Dr. Sargunadevi. A warm welcome to you and thanks for accepting our invitation and being a part of SCALE Masterclass series. She's a senior clinical embryologist. She's a competent healthcare and care professional council registered, UK registered embryologist with over 19 years of clinical experience. And she's the director of embryology and academics at the Dr. Aravind IVF chain of hospitals. And previously, she has had very prestigious positions at ARC Fertility, Kraft Women's Center at Manipal and Anpur and Prashant Hospital, and at Carey Fertility in UK. And her main interests are low-cost IVF, non-invasive embryo selection, single blastocyst transfer, and advanced sperm selection and previous IVF failures. So she's going to, talk about all about embryo grading and how we are going to, take this forward. And let us listen to Madam in the next 1 hour. And also in between, we will have some quiz and polls related to the topic. And at the end of the session, we also will have some interaction with Dr. Sargunadevi. Thank you again, and we will start the meeting.
Thank you so much for the kind introduction. Share my screen now. Yeah. Be able to see my
Yes, ma'am.
Am I audible?
Yes, ma'am.
Slides are not moving. That's one thing. It's fine. Yeah. Okay. All right. So good evening, everyone. And it's a nice evening, and there is a warm welcome from Dr. Vasundara. I would like to extend my sincere thanks to both of you, Dr. Vasundara as well as Dr. Sindhuja, thought about me to take a class in the master class. You have been doing a wonderful job, and then the presentations were excellent. Some of the times I've joined, some of the times I couldn't, but I was just watching the links and I was just watching the recordings. And thank you so much for having me here today. So my topic for today is embryo quality assessment techniques, variations and beyond. I hope I give some justice. So in the laboratory as embryologists, so we do ICSI or IVF in a minimal way in India. But one thing what excites us is the good embryo quality. When we are doing an embryo assessment, when we see beautiful embryos, top graded embryos, we are very, very happy. This is a common thing for all the embryologists. They completely believe in it. And so the success rate in an IVF program is dependent on the number of the embryos transferred, the quality of the embryos, the patient factors, the lab standards, as well as the transfer technique. So in the laboratory, in the in vitro culture, the quality and the rate of the development of the human embryos always vary. We see versatility. So each embryo is slightly different from each other. The real challenge what we have got in the laboratory is we have to lower the number of embryos. At the same time, we can't decrease the chance of pregnancy, but we have to really think about higher possibility of a pregnancy with lesser chances of multiple pregnancies. So we have to really bring in a cutoff embryo number, small number, 1 or 2, but we have to still achieve the pregnancy. That's why assessment of the embryo quality prior to the transfer becomes very, very important. Nowadays, it is 100% frozen embryo transfer cycles in most of the centers. And the cryopreservation also plays a big role in it. So embryo quality, what is it? It's defined as like an embryo is how likely the embryo is going to implant in the uterus and to give out a healthy baby, I would say. There are several stages for monitoring the pre-implantation embryo quality. What we have got is a cohort of embryos, an accurate selection with the prediction of the implantation success. With different use of different biomarkers, it would be useful, and it's the most important area of the assisted reproductive technology programs. So as an overview, I would say I'm going to cover a little bit about embryo quality assessment techniques, the traditional as well as the advanced methods, and what are the embryo scoring systems available for morphological assessment and what key factors they are looking at by the morphological grading. And how do we select embryos, and is there any relation to prediction of implantation through the methods what we apply and then select? And in the laboratory, we are surprised, not always surprised, but we see embryo arrest and development failure in some of the embryos or most of the embryos. Not all the embryos give out a nice picture. So what are the reasons behind arrest and the developmental failure, and the genetics in the embryo development, and how the environmental as well as the parental factors influence the embryo development. Also, with some artificial intelligence in the point of embryo grading and selection, how it is coming up in the recent years, and what are the recent advances in embryo grading. Probably they are very similar topics, the last two. So coming to the embryo quality assessment techniques, the conventional method is ranking by morphological appearance at specific time points of the developmental days. So this is what we do. All of us do. Some of us are doing little more, but this is what we basically do. So every day. Advanced methods will look into different things like a morphokinetics, the developmental pattern of the embryos, the ploidy, the chromosomal normalcy of the embryos, the physiology, how the metabolic and molecular pathways are going on, kind of. So we can do some assessment. Can we have some biomarkers out of it? This is what the advanced techniques do. So embryo quality assessment techniques, as I have explained, they could be invasive or sometimes, most of the times it's non-invasive, what we apply in day-to-day practice. Some of them subjective, some of them are objective and semi-objective kind of things are available. Morphological grading is the most popular one. We have PGT, time-lapse, everything is in place. Others are under research. So the first assessment technique is morphological grading of embryos. You can see the fertilized egg, the single cell is dividing in multiplying into 2, 4, and 8. And it's making more cells compacting, differentiating into a blastocyst structure. There are different scoring systems for cleavage embryos are available. Let me take through cleavage, morula, day 4, as well as blastocyst. And there are different methods. So the grading may be slightly different. There may be 4 different parameters to grade them, or 5, or 3, or something like that. Or it's numerical, it's non-numerical, whatever. But what they look at is very, very similar. So when it comes to cleavage stage embryo assessment, what they look at is the cell number and the evenness, how they divide, and the degree of the fragmentation, the presence of fragmentation and how much of fragmentation is there in the embryos. Also, we are looking at additional parameters nowadays with the knowledge from the time-lapse and the other research, that how is the symmetry, synchronicity, nucleation patterns, spatial distribution, the presence of vacuoles, cytoplasmic anomalies, etc. So cleavage stage embryos means the moment it makes a 2-cell stage up to morula, we call it as a cleavage stage. So when it comes to cell number, the number of blastomeres at a particular specific time point at the time of observation is the highest predictive factor, I would say. So on day 2, you are seeing 2 to 4 cells, it's fine. On day 3, you are seeing 8 cell to 10 cell, it's absolutely fine. If it is very slow, we always think, okay, they're not, less number of cells is not doing well. So cellular division is the main indicator of embryo viability. This is how we are assessing the embryo quality. And the embryos with normal cell number, as I have said at the time of specific timing, may be related positively to higher implantation and pregnancy rates. So recent, like last month, we had the Istanbul consensus update from the ESHRE Alpha revised parameters of like assessment timings and etc. Everything been out. So the assessment timings are slightly different. So you may have to really go through whatever previous timings and now they are. So at a particular time, at a 43 hours, what you are expecting on a day 2, 63 hours, what you are expecting on a day 3 is very, very important so that you can match up how fast or slow they are. Next comes the degree of fragmentation. A fragment can be defined as a membrane-bound extracellular cytoplasmic mass. It doesn't include any chromosomes. It's having a particular size, but then fragments are of different sizes, what we see on day 2 and day 3. So the presence of fragments in the cell volumes, how much it covers the cell volume is going to just give you the grades. For example, grade 1 can have 0 to 10%, grade 2 can have 10 to 20, 20 to 50, 50 and above will grade it as a poor quality embryo. So these are the pictures of the fragmented embryos. You can see different degrees of fragmentation. So one on the last row are having the highest amount of, the last one is having the highest amount of fragmentation, more than 50%. Certainly they are the poorest ones. So there are 2 different types of fragments. One is a definitive fragment, other one is transient fragment. Definitive ones are attached and they are present throughout. Transient may be disappearing during the embryo development. That also we need to, means if you can correlate, you can always correlate when you're checking the embryos on the next day. So the distribution of the fragments in the PVS can either be scattered or localized. And mild degree of fragmentation is okay. It can be a negligible impact, having a negligible impact on the implantation of pregnancy rates. Higher degree will always be correlated to the negative pregnancy. And the symmetry of the embryos, the cell division. So evenness of the cells, this is what we say. 2 equally sized cells, that is called symmetric division. When you see the asymmetric division, here is the picture, or uneven cleavage, you can see 2 unequal sized cells. So what happens is one of the blastomeres is large, other one is small, and the smaller one will take into next generation, means it divides into the other side, another 2 cells. So half of the cytoplasm only is been shared to the second cell from that small cell. So obviously, like they're not going to have enough cytoplasm or anything, leading to a defective lineage in the embryo. Next comes the synchronicity, the synchronous and asynchronous division, which we just say about stage-specific embryos and non-stage-specific embryos. So if the blastomeres divide synchronously, means they're multiplying from 2 to 4, 4 to 8, they're called stage-specific. And if they are of different numbers, like 3, 5, 6, or 7, not onto a different the normal multiplication pattern, we have to call them as non-stage-specific embryos. Stage-specific embryos show a better implantation rates, better quality of development, as well as the implantation rate. So this is one of the interesting structure illustrating the expected cell size of a cleavage. So you are not expecting the second cell anything lesser than 25% of the first one. So just a mathematical calculation, I would say. Next comes the nucleation. We call it as most commonly called multinucleation. The amount of information what we collect from the time lapse is giving all this information, because the static morphological grading, you may be missing seeing the nucleation. So in a cleavage embryo, presence of more than 1 nucleus per blastomere is called multinucleation. So each should have 1 nucleus, and you may be seeing it very well on the 2 cell to 4 cell stage. When they are growing, that you may not be able to see all of them because they are arranged in a different way. It has been shown to be associated with the culture media or inaccurate temperature control, especially during the oocyte retrieval. But I am sure, like the laboratory factors and parental embryo factors, everything has to be considered when it comes to multinucleation. The one on the left side, the picture is having, each one is having a nucleus, And on the right side, the 2 cell, one of them you can see prominently having multiple nuclei. So the multinucleation may be associated with high degree of chromosomal aberrations, high degree of fragmentation, uneven cell size. So it could just correlate to lower implantation, pregnancy, and live birth rates. There could be a high incidence of spontaneous abortions. So next comes the spatial distribution of the cells, and then the way how they divide, something is very important. So human oocytes are polarized and consist of an animal and vegetal pole. This is more about the embryology for clinicians. The unique pattern of the first and second cleavage divisions result in a typical pyramidal or tetrahedral structure with 4 cells with different polarity. So you can see the picture. So there is a tetrahedral arrangement. Okay. When there is a deviation from this pattern, then that could be contributing to that will be having an impact on the quality of the embryos developing. So I wanted to add this one particularly from direct cleavage. This is from our time-lapse knowledge again. So when there is a direct cleavage or a reverse cleavage, the embryos are not going to do well. This I have seen it in my own experience. I don't have a larger data, but the data, the overall data, what we have had from the time-lapse incubation, whatever I have used in the past. So we could identify this as one of the factors which could pull down the implantation rates. So when one cell is dividing into 3 cells, or any one of the cells are dividing into 3 cells, that is totally abnormal. So this influences the embryo development, even at later stages of the development. So coming to the ovoid embryos, these embryos resulting from oval oocytes, the spatial distribution of the embryos is completely different. This leads to a reduced number of cell-to-cell contacts, resulting in delayed embryo development. You can see the picture of the ovoid ones. This is a normal one. So there is a limited space for the embryos, the cells to grow, as well as lesser intracellular interactions. So compaction may not be complete, and then making a good blastocyst is not okay. So sometimes you see reasonably good blastocysts from the ovoid ones, but there could be some lesser quality embryos also will come up from the ovoid ones. So next comes the cytoplasmic anomalies, as what we see in the eggs, and even if you're seeing in the embryos, in the blastomeres, cytoplasmic pitting or cytoplasmic granularity, they are associated with reduced implantation potential, and there is a chance of they likely to be degenerated when there is a high granularity present. Coming to the vacuoles, if you see vacuoles in the blastomeres, either they are single or multiple or large, then it is going to just affect the implantation potential or the developmental potential of the embryo. Extensive vacuolization should be recorded, as it is detrimental to the spatial development. And more than 14 micrometer vacuoles, if they are present even when the division is going on, when they are developing further, then it is detrimental to the embryo. So totally abnormal development during the cleavage stage are interconnected, and even cleavage can have multinucleation. There could be fragmentation, could be chromosomal abnormalities present in the embryos because of this, causing the spatial anomalies, etc. So the alpha-Escher consensus, the revised one, has given a ranking scheme for day 2 and day 3 embryo transfer. So they have divided the day 2 embryo scoring into 3 different categories: top ranking, intermediate ranking, and low ranking. They have given the number of cells, the pattern, the cell size, fragmentation, everything is included. So we can take it as a reference for your selection of embryos for transfer in that stage. Next comes the morula stage. So morula refers to the end of the compaction process. The intracellular adherence begins, and it increases also with time. So it's considered as a good sign of implantation. So compaction as well as the cavitation. So here, these are not the best pictures, I would say. Still, you can just look at the different types of compact morula. So when all the cells are connected, you may be giving it as morula grade 1, and then partial or higher fragments, or if it's lesser, then you're going to grade them as average or 2 or something like that. Ranking for embryo selection of morula is also available from the revised one. So top, intermediate, and low ranking embryos are divided, and the specifications are given for us to just give a total grade and give a comprehensive grade to select for embryo transfer. So when it comes to blastocyst formation, we have to think about pre-compaction and post-compaction. There is a lot of different metabolic activities happening in the embryos because they are trying to differentiate into inner cell mass trophectoderm with the blastocelic cavity. So completely it is different, and there is a genomic activation as well. Like cleavage stage scoring systems, we have multiple blastocyst scoring system also are available, but I think Gardner's grading is most popular, if you all agree with me. So, and this, though it is more complex and with lower repetitiveness, it results in a high predictive value of clinical pregnancy. That's what Balaban in 2006 reported about Gardner and Schoolcraft's grading of blastocysts, released from 1999. So when it comes to the expansion degree, they have graded the blastocysts between 1 and 6, depending on the size of the cavity present in the embryo. Same time, from alpha scientists and that alpha H-ray consensus from 2011, they have given a different grading, just only 4 types. They have integrated 2 into each, whatever it is. So the thing is, like, whatever method you are following, that is what is going to give you the best results, and you need to really see it in the practice. The timing of the assessment of blastocysts is important. Previously, it was about 116 plus or minus 2 hours from the time of insemination. Now it is reduced to 111 to 113 hours for better blastocyst assessment and find out the highly viable blastocysts. It means blastocysts are faster, and then when they are faster, making it, you are expecting good blastocysts even at 112 to 113 hours. That's the thing. The degree of expansion, just a picture for you. This is the early cavitating morula. It's an early blastocyst, expanded completely, partially hatching, fully hatched. Sometimes they go collapsed. So when the consensus just giving between 1 and 6, the same early blastocyst to the hatched blastocyst. When it comes to the ICM and TE grading, generally Gardner's grading is about A, B, C, good, average, and poor, but the consensus now has introduced D as well as a 4th category. So these are the different grades of the blastocysts in one picture. You can see how an AA looks, the good inner cell mass and the trophectoderm, and the variation BA, BB, BC, CC, everything. So the additional parameter present in a blastocyst is cytoplasmic strings. It has been also mentioned in the consensus. So these are the dynamic structures present in the blastocyst, which are connecting TE and ICM cells and are involved in cellular communication. So there is a positive association from the cytoplasmic strings with the implantation rates. So this might contribute to the higher incidence of monozygotic twinning in the in vitro generated blastocysts. That is one point the author has given in the paper. So coming to embryo selection and prediction of implantation success. So how would you select the best embryos? Now you know how to grade the embryos in the laboratory. So for day 2, day 3, and day 4, we may have to just go with the ranking scheme. Whatever ranking scheme you are following, just follow it. Take the best quality embryos because we believe the quality is positively associated with the better results. But for blastocyst ranking, obviously, just not the morphology nowadays. We have got a lot of information about the early implantation, developmental features for the blastocyst, whichever is growing, so that we can collect more information from the growth. Whether it is just applicable in a non-time-lapse laboratory, I don't know. But if you have the knowledge, you can always apply. And if you 2 embryos are looking very similar, you can always use this as a tool for a deselection process. So coming to prediction, implantation prediction, any predictive value is there from the inner cell mass and trophectoderm. So ICM morphology carries a higher prognostic value for implantation and fetal development. It says, but TE is also essential, okay? And the correct way of grading ICM and TE by focusing it at different levels so that one can visualize and grade ICM and TE. This is what they are suggesting. So properly grade it. And this is one of my papers to see how, whether it's inner cell mass or the trophectoderm. There have been conflicting reports available in the literature. We wanted to see what it was. And some of my colleagues are there who really, just even attending the webinar, who really helped me with the study. It was a huge study, 2060 kid blastocysts, that's a blastocyst with non-implantation. They retrospectively analyzed the grades of the embryos and found the positive correlation with the morphological scores. ICM as well as TE, they both are correlated to the, positively correlated to the higher implantation rate. ICM, when better the grades, and ICM was giving higher implantation rates. at the same way, TE, when they are better with grades, they were also giving higher implantation rates. But TE always needed ICM to be better to give a higher implantation. Means ICM was independent and TE was more dependent on ICM to give out an implantation. So the interdependency of the TE and ICM was explained in this study. So obviously what we have to see is like better the morphology and you're getting a better result with the embryos when they are transferred. Selection by morphology, they have their own benefits and drawbacks. They're simple, inexpensive, easily reproducible. Drawbacks are mainly the subjectivity. Okay, so the subjective nature of the embryo ranking leading to intra and inter-observer variability. And sometimes the morphological markers are not enough nowadays to just predict the implantation potential or the chromosomal normality of the embryos. This is the argument. So rapid changes in the appearance during cleavage stages may result in potentially incorrect objective classification when you are doing a static image check every single day or once in 2 days. So we have to really think about what kind of advanced methods of embryo quality assessment was developed. First one is a time lapse. Second one is pre-implantation genetic testing, that's a PGT, and we're going to see little about omics technology. So embryo scoring using time-lapse incubations. What is time-lapse incubation? It's a safe and continuous monitoring of the embryo development using an ultra minimal disturbance system. Going to do the ICSI or IVF, and if it is IVF, you may have to really do the fair check and then just culture it. So anyway, from day 0 or day 1, they are getting into the culture, and then there is no opening of the slot. And every 5 minutes or even in a shorter interval, pictures are taken, and the whole video you can just really annotate and then see how the embryos, you can really play around them. So it's a non-invasive embryo selection strategy with uninterrupted culture conditions provided to the embryos growing. So the images can be assessed, and you get more and more information about the developmental pattern and timings of each event happening to each of the embryos. So there is a scoring system from the morphokinetic parameters. So the TL gives a basic score, and then I can choose the best scoring embryos for transfer, and it might be related to the higher implantation rate. So morphokinetic events. So what could you study? Anything. The time for PN formation, the time for the second polar bond, and the time for your first cell cycle, cell division, second cell division, any multinucleation formed, time for compaction, time for blastulation, etc. Everything can be studied using this time-lapse incubation. What else can be studied? Anything can be studied. Cell extrusion or the distance between 2 polar bodies. Anything you want to really just know about it. You can always take your own data and then really see whether these embryos, the implanted embryos, have done something on a particular point in time. So are they working? Because we have been having these time-lapse incubators and there have been a lot of publications on it. Do they improve the embryo quality because of the uninterrupted culture? Yes. The later reports are saying yes. Older reports were saying no. In my own experience, there was 10. Sorry. So do they improve clinical outcomes? That's the second question. Yes, they significantly increase live birth, as well as they showed better clinical outcomes all the way. And there is one of the interesting papers from 2024 from Campbell et al. There's a large number of blastocysts, 13,670, that showed clinically viable embryos exhibit a significantly faster pace of development than their counterparts. So anything which is just reaching the milestone faster, they are having a better viability and they give you better results. This is a conclusion. They've got it from the paper. Are they improving live birth rates? Of course, yes. These were some of the papers were really supporting this. And are they doing any ploidy prediction? That is another question. So it's a non-invasive embryo culture condition. And the information what you collect, can they do something about their chromosomal analysis? This is an interesting study. I always quote because I was a part of that care fertility and they were doing this. We have cultured embryos in the time lapse and they have been subjected, embryos been subjected to PGT, and single embryo transfer has been made from the euploid embryos. And those implanted embryos and non-implanted embryos, we have retrospectively analyzed their developmental pattern and we have figured out something was working as a good biomarker for us to really just go ahead, kind of. So All we found in the beginning was time for starting compaction, time for starting blastulation and blastulation timings were related to ploidy prediction. So later, as I know from my colleagues, ex-colleagues, time for starting blastulation and blastulation is just correlated to better results. So embryo quality can be assessed, even the ploidy prediction can be made from time lapse. So the next method is PGT. Pre-implantation genetic testing is just removing a single or few cells from the developing embryo for genetic analysis. So normal embryos can be transferred to increase the chances of implantation and to reduce the miscarriages. So it's an indication for PGT. So I want to just mention one slide about ploidy. Ploidy is the number of sets of chromosomes present in an organism, and euploidy is the correct number of chromosomes, and aneuploidy is abnormal, incorrect number of chromosomes. So human diploid cells will have 46, and haploid gametes will have 22. That's single end. So why do we think like these embryos are aneuploid from the in vitro culture? In practice, more than 50% of the pre-implantation embryos are aneuploid. So what I have just seen, the use of various molecular cytogenetic methods for PGT of aneuploidy has confirmed the high incidence of aneuploidy in human gametes and embryos. So obviously, in reproductive aged women, aneuploidy rate in human oocytes is 20 to 30%. Sperm contributes to 1 to 8%, and 50% of the oocytes from advanced aged women are aneuploid because of the meiotic errors. And when it goes to above 40, it's going to be 80%. So the aneuploidy rate increases with the age, with the maternal age. The contribution to aneuploidy from the sperm is very minimal. So all we understand is more than 50% is aneuploid. So we may have to test the embryos before we transfer. This is one of the methods to assess the quality and do a selection. So PGT is divided into PGTA, PGTM, and PGTSR. A is for checking the correct numbers of chromosomes present. M is for known disorders, and SR is to study the structural rearrangements. So biopsy, embryo biopsy, so can be done with the polar body and the blastomeres on day 3 or day 4 at trophectoderm. The trophectoderm biopsy is most popular now because we have enough cells for the analysis as well as we have enough cells for the embryo to just go back to the cryopreservation and later on for an embryo transfer. Embryo biopsy, you remove some of the cells and you transfer to the sterile tube and send it to the genetic laboratory. Based on the embryo PGT report, you're going to choose the euploid embryo and go ahead with a single embryo transfer, believing that it is going to give a viable pregnancy and a healthy life. Advantages of PGT is it's an additional selection tool with a morphology in addition to morphology. You can go with single embryo transfer. There is a reduced time to pregnancy. There are improved outcomes, reduced miscarriage. There have been reports. Disadvantages, we still say they are not validated yet. There can be damage happening to the embryos. Skilled labor required, special equipment is required, cost, it's higher cost. And testing platform variability was just causing the value of PGT was a little lesser because of the different platforms used in different reports. And only TE is tested and it could not be interpreting the whole embryo's chromosomal status. Coming to non-invasive PGT for aneuploidy testing. So all we can do is the spent culture media can be sent for genetic analysis instead of sending some cells from the blastocysts. So that there, the DNA can be detected, extracted, and amplified to study the genetic status of the embryo. So it can give you a comprehensive representation of the whole embryo. So the report is very similar to PGT report. You can select the euploid embryos for transfer. And coming to literature evidence, some of the RCTs, what they have performed in some of the groups, they say it's linked to the better clinical outcome with the clinical pregnancy miscarriage rates, as well as the concordance measure with invasive PGT have been reported. So coming to the advantages, they are non-invasive, no skill, labor, or equipment needed, less cost comparing PGT. Disadvantages, again, you may not do a resampling, and sometimes the DNA is not enough, and it is not very well validated according to whatever we see from the geneticists. There is one more point. Mitochondrial score is being reported from the geneticists. A mitoscore of an embryo is a number created during mitochondrial DNA testing. So mitochondria are the structures outside our normal chromosomes. They play an essential role in the energy production. They have their own DNA. They're called mtDNA. It is believed lower the mitochondrial score is associated with higher the implantation rate. The next methodology to do embryo quality assessment could be by omics. Omics will include different type of omics, metabolomics, proteomics, transcriptomics, etc., secretomics. So it depends on what particular product is analyzed. So the metabolic pathways can give us byproducts, and the metabolites can be assessed using the metabolomics. Proteins can be assessed by proteomics, and you have RNA transcripts assessed by the transcriptomics. So this is a non-invasive technique. Valuable information on biomarkers at the molecular level and the metabolic level can be assessed. So we can have an individual treatment plan. This is what they are just supporting their analysis for. So this is one of the interesting papers what I have always found. So it's Gardner's paper, and he has been doing a research for a long time with the glucose uptake, the embryo viability prediction. So Gardner's embryo selection based on glucose uptake paper to determine the relationship between the nutrient utilization by the human embryo and its subsequent viability after transfer was studied. Analysis of glucose in the spent medium was done by microfluorimetry, and higher glucose consumption was positively correlated with the higher live birth rate. So when they are making into embryos develop further on the later stage, there is a higher glucose uptake. So in future, if we have something simple as this to have an analysis in the laboratory and make a prediction that which embryos to be transferred, it will be like very easy. This is what I'm just thinking. I think this is the future. Some non-invasive technique will come across, one or more. So as per the literature update, this omics technology, we need more RCTs. They are still not validated yet. So although numerous morphological and biochemical markers have been suggested, proposed by different groups for evaluation and selection, the morphological assessment stays as a gold standard method for prioritizing embryos and blastocysts for transfer. Dr. Sindhu?
Yes, ma'am.
Can we break here?
Yes, ma'am. So thank you, ma'am, for those excellent insights. Let's continue after a short break. So we'll just have a poll now. It's like a quiz. Everybody can take part. You can see it on your screens now. Just answer. It is unanimous, just to make sure you're following. And if you have any doubts, we can clarify it with ma'am. I encourage all of you to participate. Ma'am, so can I ask a couple of doubts, ma'am, when others are answering?
Yeah.
Yes, ma'am. So the first thing is, so there are a lot of gradings for blastocysts, but like you said, most of us are following the Gardner grading system. But sometimes when we see discharge summaries from other places and all those things, they grade it as XP, BL, BT. So it is very, very variable. And then they have a superscript, subscript. So I tried reading and it is very variable actually. I'm not able to figure out where exactly this grading system is from. So I understand it is probably a hospital's own grading system. But is it universally accepted? And do you have any code, like when you see reports like that, do you have any code breaker to find out, okay, this is this blast, this is this blast?
So whatever reports I have seen, they will always say something like expanded blastocyst, hatching blastocyst.
Yes.
They never say what the quality is all about. It only tells about the expansion degree. Most of the times our hospitals do not want to disclose the information about the quality of the embryos. That could be one of the reasons why we have such things. I remember I was just trained by the Australian embryologists and they used to write XB short version somewhere from the Australia. Whether it was an approved method from Australia, I wasn't very sure because it's long back. But all I see is very less information about the blastocysts. We know they are blastocysts, that's all. And there are no pictures also to correlate what kind of grades they were. But obviously we give proper grades as well as the pictures. You can correlate. Sometimes even after thaw, they may be looking slightly different. Still okay, the original pictures will be always attached. And it gives plenty of information to the other embryologists, right? So I would suggest we all just give the proper grading of the embryos transfer so that we know what
something universal is always better.
Yeah.
So it was a nice presentation so far. So I have a couple of doubts from my end. So we were talking about unequal, blastomeres in the cleavage stage embryo. So an unequal blastomere developing into a blastocyst and an equal blastomere developing into a blastocyst. So we would want to, prioritize transfer based on that, because the grading can be good enough for both at some point. So we make a note of it in day 3, and then, we kind of prioritize transfer accordingly, or what it is like.
Yeah, means in the sense, like if you're going with a single embryo transfer, first thing you should have had a single culture. So if it's a group culture, you're not doing anything about it because you don't know which one has come out as a very good blastocyst. If you have a chance to have a single culture and you can retrospectively analyze the grades on day 3, why not? Because you are going to differentiate between these two. The evenly divided one, if it's made a blastocyst and a better blastocyst, that's over. If they're very similar, you can always go back and differentiate them so that you can decide on that. It's a very good question.
So in the similar vein, if we have two similar grade blastocysts, again, time to blastocyst also can be, decided upon the priority of transfer. For example, you said like now the cutoff is different as what it was at like 1 and 6 plus or minus 2 hours. So if you have a similar grading, we can pick up the faster embryo.
Yeah, that is a very good point, because when you check for blastocysts, grading is something very, very important, but we can't be just really going very early and doing things. But obviously, when we start the day, then we may be starting with the embryo checks. At that point, if there is a difference, then that is considered actually. It's a 4A and 3A, it's always the 4A, that's what your choice is all about. Sometimes 3BB, 3AB will be there on a, for example, this is the way we grade. So on day 5, and then 4A or 5A will be there on day 6. So people think like 5A is always better. But this is something we have to remember, that what was that on the day of day 5 check. It might be some early blastocysts or a cavitating morula. So obviously it was inferior. So this is how we just make differences between the embryos for selection and deselection.
So when we have a time lapse, we can check on all these points, but where it is not available, utilize our morphological grading timeline.
Yeah, for me, like the time lapse experience helps. I don't have one now, but the brain is always with, yes.
Okay.
It's a very good point you made.
Almost 5 minutes.
So we do encounter commonly in our clinical practice, and that can result in good grade, increase the chances of pregnancy and live birth, and especially the chances of monozygotic twinning is an interesting point for all of us.
Okay. Yeah, so what is your experience with that?
So we have few embryos, we have yet to transfer. So post-transfer, we will probably...
Let's see. For me, I do not have anything like followed up like that. I don't have it. In fact, I don't really just grade the strings or anything.
It's been 5 minutes, ma'am. So shall we end the poll?
Yeah.
About 46% have participated. Okay. Okay, some are actively participating now. Let me just give 1 more minute, please.
Yes. They're thinking probably. We have to give them time, yeah. Not as fast as you when you do the quizzes, yeah.
What are the challenges we face when we have to assess the embryo quality in advanced maternal age? Sometimes we just have very minimal embryos. Sometimes it will be a day 2 embryo, day 3 embryo. We cannot really give a blastocyst transfer.
No. Yeah.
We cannot though we know it is going to reduce the time to pregnancy. So all these challenges are there. So how do you take on this group of cases?
Advanced maternal age, if we have very limited number of embryos, the 2 early embryos, we have no other choice. We have to just transfer and see. I wish I have blastocysts for transfer for the advanced stage, but it's very challenging to leave them in the culture. But I always just relate it to a lower implantation rate, lower pregnancy rate. We just counsel the patient and transfer. Because for me, blastocyst is the option. We try to take it to blastocyst, but wherever it's not possible, we try freezing on day 3 and day 4. When there is a day 3 or day 4, it's just quite obvious that they're not eligible to go for a blastocyst culture. So that's where you are just already like putting your point in, and then it is like less pregnancy chances. We just transfer whatever is available.
If the couple is not willing for a donor program, and you're just, having a day 3 embryo for transfer, and then you probably are doing 2 or 3 transfers and it's failing, and that's causing a more emotional impact to the couple. Might
course,
as
yes.
well sometimes, we have to counsel them for, extended culture and prepare them for no embryo for transfer, because they are not even thinking of donor program. So what is the point in doing this thing?
Yeah, that is a very good point, because it depends on the patient's mentality. We give all the options. If we have enough numbers, we will just take it to blastocyst. If it is not, certainly it is not. Some patients are ready to accept the fact that they may not be having this cycle cancellation anyway. I'm just trying to say. Some of them are not, as you say. So we have to go with the patient's thoughts. As you say, like, it's a very good point that we counsel accordingly.
Okay, ma'am. Shall we move forward? I'll end the poll and share the results now. Okay. So the number of NPBs per nucleus should be present to indicate good developmental outcome at the 2 p.m. stage of embryos are. The correct answer is 5 to 7. Surprisingly, only 11% have said it, but I think now it's a good chance to know that 5 to 7. So it is not about the number of PNs. It is about the number of nuclear precursor bodies in the PNs. So 5 to 7 is the correct answer. And the buildup of ammonium in the culture media can be negated by renewing the culture medium, change the liable amino acids with stable form. So the correct answer is both of the above. 71% have said it. So that's great. And
okay, you have the page moving, or just one? First one we can see.
I'm scrolling down, ma'am. I think you can also scroll down.
Oh yeah, I'm sorry.
Macromolecule hyaluronin is added to the culture media for the following benefits. The correct answer is both B and C. So only 12% have mentioned it. So the correct answer is both B and C. That is, it helps to improve implantation rate and also helps in improving cryosurvival of embryos, because it is a macromolecule and it prevents damage to the cell membrane, etc. And when the carbon dioxide concentration is lowered in the gas cylinder, the pH of the culture medium increases. So difficult questions, people are answering easily. 65% have answered it correctly. The answer is increases.
Excellent.
The time-lapse prediction models can be used to predict all of the above. So 69% have answered it correctly. That it can predict blastocyst formation, implantation potential, and Bloily status.
I got only 3 out of 5 correct. I just did it when you were just scrolling down. Difficult question.
That is the purpose of this, yeah.
I think that
Just
the
to learn
point
something
is
new.
I mean, yeah, we're learning that that's good. Okay, ma'am. Shall we proceed, ma'am? The
Yes,
presentation.
please. Thank you.
Thanks everyone for the active participation.
Do I close this? Yeah.
You can just close it, ma'am, in your presentation.
Yeah, yes, I did. So next part starts with embryo arrest and developmental failure. And we all see in the culture, not all the embryos make it into very good blastocysts or to embryos. Freezable quality embryos are blastocysts. For transfer, later we can use it like kind of. It's always a mixture of slow embryos, poor quality embryos, good graded, average graded, and everything. It's always a mixture what we have in the laboratory. Why do we not have all of them coming out as like nice embryos for us, so that we can have a good pool of embryos for the patient? So embryo arrest and developmental failure. The developmental arrest and failure are more characterized by the failed cleavage, means no cell division after fertilization, slow growing embryos, not reaching milestones on time, abnormal morphology, poor grade embryos, or arrested development at any point of pre-implantation. So to categorize them as non-viable or as arrested, we should just check it at least for 24 hours. Then when you see a lack of cleaving activity and fail to show a sign of mitotic cellular division, we have to call them as non-viable or arrested embryos. Additionally, blastomeres of arrested embryos may have undergone degeneration partially or fully. So when we talk about embryo arrest and developmental failure, what is causing this? The irregularities in the developmental process may include slow developing embryos or as well as the embryo arrest. So there is one more hypothesis which is saying like The developmental arrest is on purpose by the natural selection processes, where the poor quality embryos are cut off, and only the survival of the fittest will go ahead developing as a babies. This is one another point being given by one of the some of the authors in one of the reports. So dividing them into early arrest, mid, and then later stage arrest. Early arrest is between day 1 and 3, happens during or shortly after fertilization. Could be because of the poor sperm or oocyte quality, be failed fertilization, no activation or abnormal pronuclear formation, be due to cytoplasmic abnormalities, the cytoplasmic or stenting defects. When it comes to mid-stage arrest, between day 3 and day 5, the embryo fails to do the transition from cleavage to blastocyst. The causes could be chromosomal or mitochondrial or epigenetic abnormalities or the abnormal in vitro conditions. Later arrest between day 5 and 6, which is characterized with the embryo reaching early blastocyst, but they don't expand or develop further. It's often due to the genetic abnormalities present in the embryos, the trophectoderm failure, or a poor ICM formation or cell cycle arrest. So when you got a table out of common causes of developmental failure, can have different categories. Could be genetic, which is due to aneuploidy, mosaicism, or gene mutations. Gamete quality, laboratory conditions, it's a vast area. Epigenetic, abnormal gene expression or imprinting disorders are present in there. The mitochondrial low ATP levels and energy failure for the embryo to go further. Or the stress for the embryos, oxidative stress or poor metabolic support. So embryo arrest is mainly due to aneuploidy. So the aneuploidy happens during meiosis, when the time oocyte and sperm are formed, or during mitosis at early cell division stage. So when considering the genetic factors, numerous genes and pathways are involved in the cycle regulation, DNA repair, and structural integrity and maturation of the germ cells, leading to embryonic arrest, which when differently expressed or the mutation is present in the genes actually. So DNA damage and methylation, when there is a misexpression of genes accountable for chromosomal epigenetic modifications, you would experience embryo arrest or developmental failure. So continuing the causes of arrest, there could be mitochondrial dysfunction. The problems with mitochondrial function, including changes in the DNA copy number, can affect the energy production in the cells and can lead to metabolic stress, potentially causing this embryo arrest. Or it could be the oxidative stress. The high levels of ROS that can damage to the embryo lead to arrest. And oxidative stress can be linked to factors like high metabolic activity in the embryo also. Coming to the environmental factors, suboptimal culture conditions during the in vitro culture can contribute to the embryo arrest. So how do you diagnose or detect the arrest? It is just one hypothetical thing. We cannot be really diagnosing or detecting early. We are just watching what is happening in the laboratory. That's it. So if you have a time-lapse imaging, you can always track back the exact timing of the arrest and how long that has been going on. By grading the embryos, you can detect the poor morphology that may precede arrest. So you may expect that. And genetic testing identifies chromosomal causes, but it's practically impossible for our patients. You can't be just testing on abnormal embryo. And the microscopic observation of whatever you document, like fragmentation, degeneration, that may be giving you an idea about what is going to happen in the next day or another couple of days for the embryos in terms of arrest and developmental failure. Is there any prevention or management strategies? It is just things like we might do, but it may not be working very well. This is what I'm expecting when you have a controlled stimulation as well as laboratory condition. Otherwise, to optimize the ovarian stimulation to have better egg quality so that we can minimize the errors, the genetic factors, so that we have less arrest, improve laboratory conditions, and see if it's a male factor infertility. Consider PGTA to select a chromosomally normal embryos. If the patient is willing, you can always choose the embryos through PGT. Avoid overhandling of the embryos so that they are less stressed. Next part is like genetics in embryo development. So the developmental process of pre-implantation embryos, where a fertilized egg develops into a fully formed embryo. This is completely controlled by the genetic information carried in the DNA. When it comes to fertilization and zygote formation, at the time of fertilization, the sperm and egg, they both contribute half of the DNA each. That's what you are seeing in the 2 PNs, forming a diploid zygote. So the zygote has a unique genome which contains instructions for the entire development of the organism. So as like a normal one, adult. So coming to the gene expression and regulation, not all the genes are on at the same time. So there is a gene regulation that ensures that specific genes are turned on corresponding to the functionality, whatever they need to do at what time, kind of. So early genes are expressed from the mother's mRNA and proteins in the egg, where later stages rely on zygotic gene expression. That's what we have maternal effect on the genes. So next comes the cell division and differentiation. So the cleavage stage embryos, they're going on, as well as they'll differentiate into blastocyst. Zygote divides through mitosis into more cells. Then we call them as blastomeres, the cells present in the cleavage stage embryos. Through a process called differentiation, the cell begins to take on specific roles. That's where we are going to think about what they are going to become as skin, muscle, nerve, or heart, or etc. So this is being guided by the gene expression patterns regulated by the transcription factors and signaling pathways. So the proteins called morphogens, they form gradients across the embryo. It's very interesting, no? Depending on the concentration of these proteins, cells can be activated. They activate the different genes, influencing what type of tissue or organ they can become. So it's like a wonderful process happening naturally and whatever we do in vitro. Yeah. So coming to the mutations and developmental disorders, these mutations are the key developmental genes can lead to birth defects and embryonic lethality. The epigenetic modifications, these are like technical terms. You may have to just understand this DNA methylation or histone modification, regulate the gene activity without changing the DNA sequence. So environmental factors can influence the epigenetics and affect the development further. Next comes the impact of maternal and paternal factors on embryo development. So what is the egg and sperm contributing to the embryo development? Certainly there is an influence from the maternal and paternal parameters so that the quality is divided accordingly. So we always mention the patient, right? We have a properly controlled program we have, and it is between the egg and sperm to decide about how they are going to develop. This is the sentence I always constantly tell the patient. We are just telling because that is the truth, and each and every patient is different. Each and every set of gametes making the embryo is very different. So coming to the factors affecting the maternal and paternal matters on embryo development is it could be age. Higher the age shows this decreased egg quality, chromosomal abnormalities, and implantation failures in the woman. Older age, there could be higher sperm DNA fragmentation and genetic disorders in the sperm. Health conditions like diabetes and hypertension can impair the embryo development from the woman, I would say. And chronic diseases, varicocele, can reduce the sperm quality. When it comes to hormones, poor egg quality and implantation failure assist if the hormones are imbalanced, and the imbalances cause sperm production issues in the men. Lifestyle, kind of smoking, alcohol habits, there can be growth restriction, developmental delays. For men, there is a reduced sperm quality. And when it comes to nutrition, deficiency can cause birth defects and growth restriction. Poor diet can cause reduced sperm count and motility. Environmental factors, if they are exposed to the environment with the pollutants and toxins, birth defects can happen. Developmental issues can be there. Toxins and pollutants can cause sperm DNA damage and cause infertility. So the mother and the father, their lifestyle, their age, the health conditions, and the exposure of them to toxins, that all matters when it comes to embryo quality. So coming to the maternal effects on embryogenesis, early pre-implantation development is clearly under maternal control, as we have seen in the previous slides, especially until the maternal to zygotic transition. So maternal age, if it's advancing, it impairs the chromosomal segregation. So there could be aneuploidy and polyploidy with the increased maternal age. It particularly, it affects the oocytes by 3 different ways. The quality of endogenous metabolite pool is affected. There is an alteration in the gene expression pattern when with an advanced maternal age. And there is a regulation of mRNA polyadenylation. So it's just 3 parameters are affected there. So coming to paternal effects on embryogenesis. So we have to really know what the centrosome is all about. That's the main microtubule organizing center. Microtubules are nothing but you can see the spindles, and the spindles are made of aggregates of microtubules. Microtubules are like hollow rod. They only organize the chromosomes, and then they will just move the chromosomes when the division happens. So they are very important. So the proper function of the centrosomes are needed for the correct assembly of the microtubules, which in turn plays a fundamental role in chromosome separation during the cell division, as I told. So if there is a problem with the centrosome and the incorrect assembly of the microtubules, there is a formation of cytoplasmic fragments and abnormal distribution of the chromosomes expected that could be affecting the embryo quality. So poor chromatin packaging could lead to the failure in the sperm decondensation moment it's just fertilizing the oocyte. So it'll be having problems with activation of the oocyte and the fertilization further. So poor quality sperm is always associated with the prolonged cell cycles and late divisions, and which may lead to developmental arrest during genomic activation. So as embryologists, we'll always look at the sperm quality as well as the egg quality, and we always try to connect that to the embryo quality development. We try to identify whether it is a maternal factor or the paternal factor. And if it matches, it's fine. Sometimes we may have surprises. Everything will look good by morphology, and we may be still having poor quality embryo development. So next comes the laboratory conditions. That's a very vast area. I think this is for a separate presentation or a class. So laboratory plays a key role in the embryo development because it's very, very crucial, and we have multiple parameters to be completely controlled to have the best performance in the laboratory for the embryos. It has to provide an optimal environment for the successful embryo development that will imitate the optimal in vivo environment. So optimization of the laboratory environment is necessary. That is completely our part. The embryologists, we have to do it. So role of environmental conditions in the laboratory influencing the embryo development. So embryo quality is certainly affected by the lab conditions. There are multiple parameters. That's what I said, like there's a big list. Still, I want to just really put some in. The lab air quality and the VOCs. So they play a very high role in terms of determining the embryo quality outside maternal and paternal factors. The particles should be less, volatile organic compounds should be very less, air changes should be appropriate, air quality should be perfect before we think about like why something has gone wrong in the laboratory. Ambient temperature must be between 20 to 125, and humidity should be appropriate, and so that we have the normal functioning of the equipments in the laboratory. Incubator types, different incubators. You have a box incubator, you have a benchtop incubators, you have a pre-mixed gas supplied, or you have they are mixed with two different gases, and the gas quality, are they medical graded, and what is the level of the gas at the time you are just culturing these embryos? Everything matters. Culture media composition. So there are different types of media available in the market, and we are using single step or sequential media. And we may refresh them on a particular day, may not. There's a big variation in the SOPs in every laboratory that all may influence the quality of the embryos developing in the culture incubator. The temperature and the pH is very, very crucial during the time of the incubation. So the temperature of the incubator, as well as the pH of the culture media at the time of incubation, as well as the temperature control of all the equipments in the precise range is very, very important. Are you using a high oxygen incubator or a low oxygen incubator? That could be just making possible differences. Light exposure or exposure to anything else, to more human beings, high VOCs and everything has to be considered. Embryo handling and micromanipulation. So how long we are just taking the embryos out and putting it in. It's a few seconds can make the spindles disassembly, as well as they take really longer to have a reassembly after you keep it back in the culture incubator. That's what we have to really remember, and how quick we can make it, and what kind of media we are using during the manipulation time, and how the temperature and everything is maintained in the equipment during the time. And the embryo cryopreservation, because fresh transfer is very different from frozen embryo transfers. We are subjecting the embryos into a new process where you are just stopping all the metabolic processes by introducing different chemicals, getting it back. Though we are getting very good results from the frozen embryo transfers, it will still have its own damages. So it will all the list I have for, or even more, you maybe always discuss about it in the question answer session. What I say is, like, the laboratory with the proper equality and the good choice of equipments with the thorough knowledge and well controlled with the proper QC system in place, and like well written SOPs for all the processes happening in the laboratory, and it's reviewed regularly, and you have a non-conformity system in place, and you have a risk assessment system in a place, everything is important. So this is all going to tell you like how good you are, how well the lab is controlled. So we can minimize the influence of laboratory-oriented damage to the developing embryos. Okay, like this said, there's only one thing that's truly important in an IVF laboratory, that is everything. So we have no excuse. We may have to look at everything in the laboratory. Coming to the recent advances in embryo grading, I would like to talk about artificial intelligence and how we are using AI and machine learning ML in embryo grading and selection. That is, I thought it was very latest because PGT for more than a decade, TL very similar, but AI is just coming up. So standard morphological grading is gold standard. That's what I have said, but it's subjective. So AI can enhance the selection process, but being objective, and the score can predict the implantation rates of the embryos very well. That's what's been the recent papers I'm seeing. So these are the pictures, just to see like nothing different because we all talk about AI. What do? We do nothing. We just do the same culture in the laboratory, and the AI software will be installed. And then it may be looking into the images of the embryos in a better way. That's what they claim actually, because we may be seeing it in the 2D, maybe looking from a different plane so that there is a better observation of the parameters present in the characteristics of the embryos. So if it's a normal morphological grading, this AI will give a score and you can correlate with your morphological scoring, and better the scores, the higher the chances of them giving an implantation. So you have to choose that. If it's a time lapse plus AI, then the time lapse is going to give a basic score as well as the AA score and your morphological score. So you have a comprehensive score for an embryo to be selected for transfer. The recent systematic reviews and meta-analysis, so I'm just reading some of them. So what are they saying? Higher predictive accuracy between AI versus embryologists. First thing we need to know, are they competing with us? So are we better or are they better? But they have said like higher predictive accuracy because they are just looking at things very different. Better ploidy prediction than PGT, that's a question. The latest report is saying no, that's a systematic review. And AA potentially will improve. There are gaps that has to be filled. And we have to really look forward to having better results with the AA. This is what the recent report from 2025 review saying, as well as there is another paper from 2022 saying the same. So AA has its own future. So what are the benefits of AI in embryo grading and selection? So there is an increased accuracy and consistency, and it's objective as well as it does reproducible assessments. There is no difference. So there is a potential for better outcomes, and number of embryo transfers can be minimized. So in a shorter period, you may be just giving a good result. And time efficiency for embryologists. So we don't need to really sit and do that job, then they will be doing it. And so it saves our time. That's what they're trying to say. But that is our job. Personalization by integrating patient-specific data. So AA can work on everything, not only embryo grading. It can work from the patient history, the stimulation, the treatment plan, throughout the treatment, as well as it can look into any part and it can connect things together and give a score for you and a suggestion for you. So when AI is integrated with time-lapse incubators, that works very well because they can automatically look into the embryo pictures. So what is a predictive ability on clinical outcomes from this artificial intelligence? It's a very interesting paper from 2022. This huge number of time-lapse monitored embryos, that's 63,000 plus. The machine learning model rapidly and automatically generated annotations. So when you are using time-lapse, the time you take for annotations is very, very long actually in the laboratory. We actually make somebody sit and then do that. They have to annotate, means they have to really just fix it. Just go back and see this is a 4-cell time, fix it, what 8-cell compaction, whatever, and anything extra you see, you have to document and then manually annotate all of them for all the embryos, for all the patient present at a time in the time-lapse machine. So this AI will do all the annotations like that. You don't need to do anything else. It's going to give you a score. You can choose the embryos. You can do your own grading, but you can compare with that. So efficacy and comparability of the ML model to automate reliable, utilizable annotations was demonstrated. That's the main advantage of having AI with time-lapse. So using this ML model, they have got the predictability for live birth. It is measured and validated. So we can utilize it for the future patients culture in the time lapse with the AI application. Very interesting. So this one is improved clinical outcomes been demonstrated using the scores applied from the VitroLife IDA scores. So you can see the graphs, and the higher the score, better the implantation rates, live birth rate as well. So the AI, is it useful in ploidy prediction, the time lapse? So this was again 2023 interesting paper, comparison of 12 machine learning models done from 2012 to 2020. It's a long 8-9 years. 9 IVF clinics been just put together, data from 9 IVF clinics put together. 1,725 patients of time lapse culture, it's a large number. 8,147 PGT embryos. So they have done time lapse culture, AI was applied, that is ML, and PGT embryos been assessed. So what they found was all morphokinetic variables were significantly delayed in aneuploid embryos. As I've just told, faster is better. No association with ploidy status for morula or cleavage stage. So nothing. So when it comes to blastocyst, it works very well. Predictive markers were time for expansion and T grades. That's what they found, time for expansion for blastocyst. And T grades, I don't know, like we may just think about ICM more, but this is what the paper is from all the clinics. So deep learning model was superior than morphokinetic and only morphology model selections. So they found ML was working better in hand with morphokinetic and morphology model, comparing when they are working differently, separately. So artificial intelligence with TL. So are they really working on this? Embryo assessment and selection, that is our current topic, what we are discussing today. Yes. Ploidy prediction, it looks like they are helping with the scores. Pregnancy prediction, yes. So they are on to every area. So benefits of AI integrated TL, again, the workflow efficiency. If somebody is annotating, the AI is annotating, I have a lot of time to do other work. So there's a diverse data to minimize bias. Clinical protocols and patient profiles can all be just compared using AI, and they can give one approximate score or suggestion. Better prediction of implantation and live birth using this AI, and analysis of many viability markers are possible using this. They can look the embryos into different planes, and they can just identify something novel and interesting, comparing the ones what we are having now. So what are the limitations of utility of AI in the laboratory? So we need to have robust, diverse training data to avoid the bias. Validation should happen across different clinics and populations. It has to come more, like we have to collaborate, and then they have to produce more and more data on the utility of AI. And the regulatory oversight and ethical concerns to be considered. Lack of transparency, that is one of the limitations. They're using black box models. Sometimes we don't get the exact information like our media composition. So that's what they say. It's one of the limitations of the utility of AI. So we have been getting reports very recently. We are getting into the next breakthroughs other than ICSI and vitrification, I'm guessing. So Mexico has reported a successful live birth from a robotic ICSI. So AI ICSI baby is being produced. That's the world's first baby. And there is one from New Zealand that is also from April 25, world's first IVF baby using AI. The AI has fertilized, they have not said what it is, fertilized, and they have assessed the selection possibly. So these kind of reports started coming. These are the recent advancements. I'm just thinking when it comes to embryo grading and selection, AI is helping. So image analysis, that is one of the important areas we have to really talk about now. There have been just beautiful presentation in the international conferences that how it was like really fantastic, like how they do the imaging. We don't even need to do anything, but it gives different colors to identify the ploidy of the embryos or some abnormality, so that we just see it's already there, and then you're going to choose depending on how the coloring were. So this one is automated identification of blastocyst regions at different developmental stages. So automatic regional segmentation of the discrete regions of a blastocyst, I will have to just read, using a digital microscopy images could aid in overcoming the drawbacks of the historical methods by increasing the objectivity and reproducibility of the embryo selection process. So they are colors. You can see there are different colors given for different things. and that's going to give a overall score based on the imaging. So that is really helping. So based on the intensity patterns of the regions in the images blastocyst is given, it is possible to automatically infer the embryo quality or its potential by using the artificial intelligence. So the embryo selection and the prediction of implantation, that is one of the things you have asked me. So this is what is being done by the artificial intelligence by giving different colors and intensity. So it's an automated identification of the blastocyst different stages. This is one another paper, very similar, but they have used a different parameter to study automatic measurements of the embryo morphometrics using AI as a prediction tool for implantation potential. Those say they have measured the diameter, the perimeter, the size of the inner cell mass and everything. So it is helping in identifying the best quality embryos with a higher implantation potential so that we have a better success rate. So then this is the end. And as a conclusion, we can say, to summarize, assessment of embryo quality by different methods are available for selection of the most viable embryos for transfer. Morphological scoring is the classic method what we all use, but we all have to really upgrade ourselves and gain the knowledge from other methods also. Non-invasive prediction of viability is on focus because we may have to have something non-invasive or more so that we can just get on with identifying the viable embryos very easily. That just makes our job really easy. The utility of AI and ML algorithms in image analysis of embryos are under validation. So pre-trained AI models demonstrating accurate and reliable performance on euploidy ranking and live birth advance prediction is the future. So what are the future directions of AI? Is integration with multiple data, as you have said. So it can collect all the information, morphokinetic data, genetic data, transcriptomic profiles, and also the AI's its own scores with the patient profiles can be collected together to give out the suggestions from AI. And real-time AI guidance during embryo biopsy or cryopreservation is possible in the future. And we will have more personalized algorithms accounting for maternal and paternal factors also will be available for us in the future. So this is the end of my presentation. And these are my other references other than the ones I have mentioned in the slides. Thank you very much for your patient listening.
Thank you so much. Your references are like, volumes, and you've concise, references in 1 hour. Thanks for that exhaustive lecture. Actually, you need a voice break for 2 minutes. So Sindhu will put the poll questions, and then we will just take 2 questions before we, close for the day.
I have launched now. I kindly request everyone to participate. Ma'am, thank you so much. You spoke non-stop extensively. And I still remember when I emailed ma'am for the content, she said, ma'am said, this is like 5 different topics in one. But you so beautifully compiled everything, ma'am. Thank you so much.
When I was making the slides, I was understanding it more like it's really like important to know the in and out of the embryo development and the quality and what we are doing and then what is the future kind of. So thank you so much for the opportunity for me to just look into this.
No, ma'am. And actually, I think the main thing is like in conferences, we just get to hear a gist of like everything. But when we know in detail, it really adds to how much we can understand comprehensively, actually.
That is the purpose of your masterclass. Yes, amazing.
No, such important, whether it is one single point which you said, you expose the, oocytes for a longer time, the